Hi, you guys. I wanted to put together a video for chapter 17 to help you walk your way through the PowerPoint slides that are available to you on my website. So this one is chapter 17, from gene to protein. Let me hide this really quick. Your list of must-knows for this chapter are obviously the key terms like transcription and translation, and we'll go into gene expression a little bit here as well. You need to know the major events of transcription, so that's going to include initiation, elongation, and termination. The difference between eukaryotic cells and prokaryotic cells with regards to this process, the steps of translation, and how point mutations can change the final product, your protein product. In other words, if we change codons in the DNA or change nucleotides, how that will result in differences in amino acids or the final protein product. So let's just start at the best place to start, the beginning. 17.1. This is all about gene expression, which is just a fancy way for saying how DNA gets read, or the process by which DNA directs the synthesis of proteins. And it used to be thought that every gene that you have codes for a protein or an enzyme, right? So you have a gene for the enzyme that digests milk, right? Lactase. There is a link in this PowerPoint to like an animation that explains the experiments done by Beadle and Tatum and how they proved that this wasn't actually accurate. That it's more like not just one gene that codes for an enzyme, but that there's a pathway, that genes code for enzymes in a pathway and eventually can lead to a final product. So now we know it's not necessarily that the gene codes for the product, it's that a gene codes for a polypeptide, which is going to be used in order to make a final protein. So the polypeptide chain will probably have to join up with other peptide chains that might be made by other genes before it actually is useful in a cell. Now that we know more about RNA processing, we have a more accurate model of a gene. Now we say that a gene codes for an mRNA molecule. Now that RNA can code for a polypeptide, but we can actually change the way that RNA leaves the nucleus, which can make the final polypeptide different. So all we know is that a gene codes for RNA, but the fate of that RNA is still subject to taking different paths. The flow of genetic information is also known as the central dogma of biology, and you'll hear it called that or see that in print often. The central dogma is basically this set of rules that the universe lives by that helps to explain why everything works the way it does. So the flow of genetic information, or the central dogma, the central idea that is necessary to make everything else make sense, is that DNA gets changed to RNA, and that RNA then codes for a protein. This is the flow of information. DNA gets passed from generation to generation. Those proteins get acted on by nature, and you have the big idea of evolution, driving the unity and diversity of life. But it all hinges on this concept, that the DNA is the code, but that it has to change in order to make the final product that nature can then act on, which is the protein. The site of all of this fun action is at the ribosome. That is what we call the site of protein synthesis. Here's just a graphic from your textbook. It shows prokaryotic gene expression on the left, or the central dogma, flow of genetic information, and eukaryotic cells on the right. What I'm hoping you notice by looking at the image is the simplicity of the prokaryotic cells process DNA, mRNA, polypeptide chain. Look over at the eukaryotic cell and you can tell that there's a location difference. There's some stuff happening in the purple circle. That's the nucleus. Transcription happens there. We have an extra step called RNA processing. Then translation happens in a different location in the cell, the cytoplasm. So under this assumption that one gene equals one RNA molecule, we have to go back and review what is RNA, right? How is it different than DNA? So DNA, remember, is a nucleic acid composed of nucleotides, but so is RNA. DNA is double-stranded, but RNA is a single-stranded molecule that has everything to do with its ability, its function. The DNA is made up of a deoxyribose sugar in its nucleic acid, whereas RNA uses a different sugar. Not that important, but it is a structural difference. DNA has a thymine as one of its bases, whereas RNA has uracil instead. The others are the same. So the A and the G and the C are the same. DNA, remember, is a template for everything that the individual can do and all the proteins that it can make. DNA itself doesn't do any of it, but it is a template for RNA that can do lots of things. And by lots, I mean more than what I can hold on this slide. RNA plays many, many roles in a cell. There's pre-mRNA that you got from the POGIL today, the precursor to mRNA. There's mRNA, which is the edited version, how the code leaves the nucleus. There's transfer RNAs, or tRNA. Those, their job is to take amino acids to the ribosomes so that they can be put in the proper order. We have rRNA, which stands for ribosomal RNA, and that's what the majority of the ribosome itself is made up of, is a bunch of wrapped up, wound up RNA, but it's wrapped up in proteins. Then there's snRNA, small nuclear RNAs. They play a role in the spliceosome, which we will talk about when we talk about RNA processing. Then we have srpRNAs, and they also play a role. We'll talk about them in a little bit. RNAi, very cool, a more recent introduction to the RNA team. That's something we just recently discovered. Very cool, and we'll talk more about it. And then ribozymes. Ribozymes are RNA, but they are wound up with these enzymes, and they act as an enzyme, but they have binding sites that are controlled by RNA. So again, when we say the genetic code, we're talking about how the letters, the order of the letters in DNA actually translate to proteins. So there's going to be half of the DNA. When we say half, we mean one of the strands, one of the sides, is going to act as a template for synthesis of the RNA. It is going to build in the 5' to 3' direction, as it always does, because polymerase can only add to the 3' end. And the mRNA is then read in triplets called codons, which is 3 letters of the RNA that code for only one amino acid. This is a codon chart. We taught you how to read it in regular biology. Hopefully you remember some of that, because we'll put those skills to good use again this year. There are 64 different codon combinations, and a lot of them code for the same amino acid. The reading frame means that the groups of 3 need to be read in the correct sequence in order to end up with the final protein that you're supposed to end up with. So hopefully you're thinking already about how a mutation might change those triplets and change the reading frame. This code, again, is universal. Every single organism on the planet uses the exact same code. ACU codes for threonine in me. It codes for threonine in you. It codes for threonine in the blade of grass outside. it codes for threonine in prokaryotic cells. Anytime A, C, and U are together, they always code for the exact same amino acid. More evidence for the unity of life and evolution. Into 17.2 focuses mostly on transcription. Transcription, again, is a transcriptional unit, is a segment of DNA that codes for a specific RNA molecule. So we have, again, all the RNAs are coded for by your DNA, even the transfer RNA and the ribosomal RNA. All of those come from your DNA, and it codes for how to make them. There's an enzyme that builds all of these RNA molecules, again, including messenger RNA, tRNA, rRNA, snRNA, RNAi. All of those are built by the enzyme RNA polymerase. RNA polymerase separates the DNA. It elongates the DNA very much the same way as DNA polymerase does in the 5 to 3' direction, and has uracil as one of the nucleotide bases that pairs with adenine instead of a thymine. RNA polymerase is attracted to the start of a gene by what's called a promoter sequence, and then it knows when to stop when it reaches a terminator sequence. So let's talk a little bit more specifically about those. RNA transcription, or the making of it, can be broken down into 3 parts: initiation, elongation, and termination. Initiation is very simple in a bacterial cell. The RNA polymerase is floating around. It will attach to the start of a gene at something called the promoter sequence. And then it just goes, right? So going back a slide, it just reads in that direction, building 5' to 3', reading 3' to 5'. Initiation in a eukaryotic cell is just like everything else in a eukaryotic cell, more complex. So initiation in eukaryotes starts at something called the Tata box. I did not make that up to try to be funny. It really is called the Tata box. It is a DNA sequence where we have a repeating bases, TA, TA, AAA. So wherever that is found, that signals the beginning of a gene. So it's called a promoter sequence. It's a little bit, the Tata box is a region a little bit upstream of the promoter. So when the transcription factors bind, they form kind of like a clump or a cluster, and that will call RNA polymerase over to bind. So in order for RNA polymerase to even know where a gene is, the transcription factors have to first bind to the Tata box, and then the entire promoting region gets activated, and all these other transcription factors come to the party, and that signifies RNA polymerase to come over and bind. So once the transcription factors are there, RNA polymerase binds, and you can see in the third picture down there that will then initiate transcription. Elongation is the part where the RNA polymerase is putting the RNA nucleotides on the DNA. So it's adding RNA to the 3' end of the growing strand because it's a limitation of polymerase, because of the where the carbons are on a nucleotide and where the energy will be when those phosphates break off. Elongation just moves along the DNA, untwists it, then it just allows it to rewind on itself as soon as the mRNA is made. And then we have to cut the RNA loose. So it is binding, just hydrogen binding, so loosely binding to the DNA. But as soon as the RNA reaches, or the RNA polymerase reaches a termination sequence, it knows to disengage. So it will come across this chemical signal that tells it to release, and then the RNA can float away. So it's called a terminator sequence. It doesn't have a clever name like Tata box. It's just, again, repeated subunits of DNA that tell the RNA to leave. So now in eukaryotes, we have another step to do. In prokaryotes, this is done. So the mRNA now gets read by translation. In eukaryotic cells, we have to process it still. So this is called pre-mRNA. We are staying in the nucleus for this process. Remember, in prokaryotes, there is no nucleus, so there is no time to process the RNA. It is ready to read. It goes straight into the next process, or straight into translation. But let's talk a little bit more about what's going on in eukaryotic cells. So the pre-mRNA is still hanging out in the nucleus. It is not ready to leave yet. We're going to add what's called a 5' cap. That gets added to the 5' end. It's basically a modified guanine that will end up helping the RNA find its ribosome. Then we have something called a poly-A tail. Poly means many. A refers to adenine, the base. So a poly-A tail is basically a whole bunch of A's get added on to the end of the pre-mRNA. Anywhere from 50 to 520 or so adenines are added. The distance or the length depends on how long that protein will be needed for. So if it is a short-lived protein, meaning like we only need it for a short amount of time, then we'll put a short poly-A tail on it. If this protein is going to be needed for a while and we need to keep making it and the transcript needs to be longer lived, then we will add a lot of A's on it. So that will help you figure out what the A's do, right? It has to do with its lifespan. Again, the 5' cap is going to help get it out of the nucleus. It'll help it get through a nuclear pore. It's attracted to a nuclear pore. It also is attracted to one of the binding sites on a ribosome. So it'll help it dock on a ribosome to get started with translation. But the poly-A tail is specifically there to protect it from degradation. There are enzymes that recycle RNA, and they are right outside the nucleus door. So as soon as that thing gets out, there will be enzymes trying to chop it up and degrade it. The poly-A tail is kind of like a telomere for the RNA. It protects the important code while the rest of it gets digested. This act of changing the RNA into different codes is called RNA splicing. So 5' cap, poly-A tail. Then we have splicing. That means that portions of the RNA can get cut out before it leaves and get spliced together. If you look at this picture and the model from your POGL, so I have that one nearby, this guy right here. So this model is showing you what parts of the pre-mRNA are removed. And it's these segments called introns. So the introns are non-coding sequences, at least not for this protein. So we're going to remove them and splice together the exons. The way I remember which one gets cut out is I remember that exons get to exit. So the introns stay in the nucleus, right? They get removed, they don't get to exit. The exons are the part that leaves the nucleus and goes on to be translated. Those are the coding parts of that specific RNA. So the introns get cut out, exons splice together, and now it can leave the nucleus. A little bit of detail about how that happens. It's called RNA splicing. And this is where we get into some of the other RNA accessories like SNRPs, small nuclear ribonuclear proteins. They're abbreviated SNRPs, and we call them SNRPs whenever we're speaking of them. So we basically take these small nuclear RNAs, which are sequences of RNA, and we combine them with specialized proteins and then put them in a specific conformation or arrangement so that they will complementary base pair or attract to certain parts of the RNA transcript, of the mRNA, the pre-mRNA. So once it recognizes where those splice sites are, it holds the RNA transcript there, and then it cuts or breaks pieces of the RNA out. So if you look in the picture on the right, you can see a spliceosome. That purple blob is made up of many SNRPs. It's made up of 4, in this case, 4 SNRPs. And each one is specific to a certain sequence of RNA. Now, you can cut in different places depending on your SNRPs. So you can use different small nuclear ribosomal RNAs, and it will attract to different sites and cut different places. So we can take one mRNA transcript and cut it in a whole bunch of different places and make a different mRNA. So this is kind of cool. A spliceosome is an example of a ribozyme where RNA is actually working together with a protein like an enzyme to catalyze the breaking of RNA. Catalyzes a reaction. Kind of cool. And since we have a bunch of different ones and we can cut DNA in different places, that then brings into mind the function or purpose of these non-coding sequences. Some of them are non-coding for proteins, but help to regulate other gene activities. Like they may not code for a protein, but they turn on a gene somewhere else. Or they slow something down. But more importantly, the function of introns is you can have one DNA code for multiple proteins or multiple amino acid chains. All you have to do is change the way you splice it. So because there is alternatives to how you cut the RNA, you can make the same RNA template, the pre-mRNA, but process it differently. Cut it in different places, and then the final copy is a totally different, unique sequence. And you can make a different polypeptide each time. That means not only does one gene code for a polypeptide chain, it can code for more than one, which means that we have a lot of genes, but we can make even more polypeptides, which gives us a huge library of proteins that we can make. So with small amount of genetic information, we can be as complex as we are and make so many different proteins, even though we only have 20,000 genes. We can make way more than that when it comes to proteins. Very, very cool. Translation.